Aha.
All right. Oh, I see. The gates are opening.
Mm-hmm.
Let's just give it a couple of minutes for this to populate. All right, but we passed 50, so maybe in the interest of time, as people join, we'll get started. Welcome, everybody. My name is Mitchell Warren, and I direct AVAC, and I'm delighted to welcome you to this webinar, the first in a series, but not the first of today, to focus on HIV vaccines. I think as you all wouldn't be here if you didn't already all know that today is HIV Vaccine Awareness Day, and it actually marks the 25th anniversary of a speech that Bill Clinton gave at Morgan State University in Baltimore, Maryland, a commencement address where he talked about the 21st century being the century of science and a focus on vaccines and vaccine discovery. And he certainly couldn't have imagined then in 1997 what would actually happen in the 21st century, both of the wonders of vaccine science, but also of the challenges of pandemics, ones that have been with us for too long and continue in HIV and TB and so many other things. And certainly now for the last 2.5 years, COVID-19. Perhaps the most remarkable result of that speech was not HIV Vaccine Awareness Day, as important as today is. It actually was in the establishment of the NIH's Vaccine Research Center, which was an outgrowth of President Clinton's speech that day. And I think for those of us at AVAC and in the field of HIV vaccines, we think about that a lot, sitting here and knowing that the VRC pioneered some of the work you'll hear about on this call today in mRNA technology that got us so quickly a COVID vaccine. My task, though, is super simple, and that is to welcome everybody. A quick run of show. I'm going to turn it over to the colleagues you see on screen for the next hour. We really hope that it is interactive. We want to encourage you to use the chat feature as well as the Q&A. And my dear friends and colleagues, Stacey Hanna and Ntando Yola, are going to moderate those conversations and bring us into community together to think about where we are and where we still need to go in the search for an HIV vaccine. It is being recorded, and we'll be posting it along with a number of other HIV resources on the website. I should say, if you go to avac.org/HVAD, not only can you see the recordings and the materials, you can also actually watch President Clinton's speech from that day, which really is quite a remarkable one and worth a historical moment for people in your spare time. But now I have the great pleasure of introducing one of the people that not only co-founded AVAC, but actually taught me so much about vaccine science and vaccine advocacy, and that's Bill Snow. As all of you will know, Bill co-founded AVAC now almost 27 years ago, went on to also lead the global HIV vaccine enterprise, and we're delighted that Bill continues to serve as a senior advisor, not because of age, but because of stage and what he brings to the table. And we're delighted that Bill is serving as really the convener of this series of 3 calls that start today, and we have one each of the next 2 weeks that Bill will describe and then open up the rest of the call. So welcome all, and Bill, over to you.
Thank you, Mitchell, and welcome everyone. This is the first of 3 weekly webinars. I recommend that you watch all 3 of them because we're actually triangulating around one complex problem, and no one call will give you the whole picture. They have titles that are striving for alliteration. They all start with P's, but I'm not sure they differentiate the topics all that well. Today we're going to focus on platforms, however. What we're looking at is what AVAC has called the Miracle of Rna. In terms of COVID, it has indeed been a miracle, but I think people are have higher hopes for its ability to help us with HIV than, as I think you'll hear today. It will help, but it won't solve the biggest problems. And there's also a very nice webcast that you can listen to from AVAC that covers the basics of the differences between antigens and delivery systems, which I think is a really good starting point. Next week, you'll hear about what they're calling processes, which is essentially how we're going to test various candidates. The candidates are largely being made by RNA, but how we test them is also changing. And then the third one is called prospects, and that's going to sort of circle back and talk about the failures of some previous large efficacy trials and what we learned from them and how we're moving forward. So just as a bit of a preview, I want to say that we're, at least in my mind, we're moving into a new phase of HIV vaccine research, and in some ways it's a more enlightened phase. We now have the ability to test more things more quicker and more precisely, and I think that the field is taking advantage of that. Originally, we were hoping to have a vaccine in 10 years. We got a COVID vaccine in 1 year. We're now on our fourth cycle here. And I think that what has changed every 10 years has been the paradigm for how to find an HIV vaccine. And right now, we have the results from the AMP trial that are making people excited about the potential of broadly neutralizing antibodies. And we have the ability now to do more of what you'll hear this word a lot, iteration. And that's, in a sense, that's the practical solution. The practical solution is that we can run trials faster and smaller and get partial answers about whether we're on the right track or not. And so that's what I see as the new phase. You'll hear more about it from Nina and Bart. The other thing that I've been asked to talk about is advocacy. And I do think that our role is changing as well. Since there are going to be not just a few big, gigantic trials where we wait for years for the outcome, there's going to be a long string of multiple trials, some of them in parallel. And they're all going to be small, and they're all going to require volunteers to be willing to participate in essentially exploratory trials, in trials where we try things out and see how they work and see if they're taking us on the right path. So I am encouraging everyone to tell their friends that they should register with the HVTN and the other groups that are about to do these trials so they can enroll as quickly as we can run them. And also to continue to advocate for the urgency of an HIV vaccine. So I'm going to now introduce our first speaker, who is Nina Russel. She's the Director of TV and HIV Prevention at the Gates Foundation, an old friend and wonderful colleague. Nina, over to you.
Thank you, Bill. I am thrilled to be here this morning, early on the West Coast on HIV Vaccine Awareness Day, to kick off an important day with all of you, and to talk a bit about first big picture perspective from the Gates Foundation's side, and then a bit on the platform piece that Bill mentioned, specifically RNA. Levi, if you could go to the next slide. I wanted to start by putting our HIV vaccine work in the context of our broader HIV strategy, so that everybody understands sort of where it fits in terms of the overall Gates Foundation investments and thinking. So the overall vision for our program is to accelerate what is already an ongoing reduction in the incidence of HIV infection in high burden geographies and populations. with the ultimate goal of achieving sustained control of the epidemic. And we approach this from two sort of broad perspectives. One is focusing on reduction of onward transmission, and the other is preventing new infections. And in terms of reducing onward transmission, the emphasis is, not surprisingly, finding people living with HIV, particularly hard to reach groups, and linking them, testing them, getting them diagnosed and linking them to treatment. And then once treated, maximally and sustainably suppressing virus load and maintaining lifelong antiviral therapy. On the prevention side, we work both at the delivery end and of course with a significant focus in R&D. And in terms of delivery, we look at both promoting scale up and sustainable coverage of existing prevention interventions such as voluntary medical male circumcision, and looking at innovative prevention service delivery approaches, particularly thinking about how we can deliver new PrEP interventions. And then in terms of R&D, and you can click thank you one more time, we focus on development of long-acting PrEP, as well as, of course, vaccines, which is the topic today. So that should just give you some perspective of where vaccines fits into the overall Gates Foundation HIV strategy. Next slide. I wanted to mention that for the past 17 years, really the duration of my employment at the Gates Foundation, our work has been organized and funded through a model we call the Collaboration for AIDS Vaccine Discovery, which some of you are certainly aware of. And for us, this is very important because it's not just a grant funding mechanism, but it's a way of organizing the community that we fund, such that we bring together scientists that are working in the early and late discovery space who have novel concepts. We fund them ideally as collaborative groups. And we link them to what we call central service facilities. So these are really best-in-class, centralized services that support performance of assays, such as T-cell assays, antibody assays, support evaluation of vaccine concepts in animal models. We have a couple groups working on innovative mouse models, and very state-of-the-art statistical analysis. And then importantly, linkage to product development support. So resources that really help advance novel concepts into first in human evaluation with the eye towards potentially developing vaccines as products. And we support all of this through alliance management functions and really emphasize sharing of data, early sharing of data, collaboration, and sharing of materials as well. And all of that happens via a web portal. So this is an important mechanism that the foundation has used, again, for many years. Next slide. So our vision overall for an HIV prophylactic vaccine is broad coverage with a durable, high efficacy vaccine that will ultimately transform the epidemic by reducing incidence. Okay, this is a vision, something we will all someday celebrate when it's achieved. The goal related to this is development of a vaccine that's safe and elicits a coordinated and comprehensive immune response. And I'll focus more today on what our ultimate focus is, which is the elicitation of potent and sustained both humoral and cellular immunity capable of overcoming the very challenging HIV sequence diversity that we're seeing by preventing or potentially rapidly aborting a primary infection. Next slide. So I want to mention that we do have a target product profile that we use to guide our investments. And for us, this doesn't mean that we're only funding potential products. We do support research. We think that discovery is very important, and answering fundamental questions is very important. But what it does mean is that what we do support is with an eye towards the potential to develop a product, and we make an effort to try to think through what the path to a product would be for the various things that we're supporting. And so, for example, and the TPP includes both minimum criteria for what would be minimally acceptable as well as what would be ideal. So, for example, a minimal target efficacy of at least 50%, a duration of protection of a minimum of 3 to 5 years, a vaccine regimen that can be delivered within a 6-month time period, and stability, shelf life, access, and cost parameters that would be acceptable for deployment in lower middle income countries, which is absolutely critical and a requirement for the Gates Foundation and the approaches that we support. Next slide. So I want to talk a little bit about where I think we are, because this is an incredibly important time, as I think has been said already this morning, for us as a community to reflect on HIV vaccines and where we are in our learning. And I've been thinking of it as both a virtuous and a vicious cycle of learning of late, because, virtuous because I think as we obtain some results that are good in that they teach us something, and that those results enable us to move forward to hopefully get more good results. I think we're also seeing that as we learn, as we answer one question, it leads to new questions and new problems that we need to solve. And over the past several years, as we've all been learning by watching COVID evolve and develop and the remarkable progress of COVID vaccine development, the HIV vaccine field has experienced the readout of the 3 largest efficacy programs that have been supported really in the last 5 to 8 years. And that was first the HVTN 702 UHAMBO trial looking at the P5 program pox protein prime boost, the AMP trial, which was mentioned, looking at intravenous delivery of the VRC01 CD4 targeting monoclonal antibody for passive administration, and the HVTN 705 Imbokodo trial looking at the Ad26 protein prime boost. So these were all really seminal trials that read out critically important results for the field that of course have led to a number of questions and a number of learnings. Next slide. So we've learned a lot, as I said, from these studies, and we're continuing to learn a lot. There's ongoing immunoanalysis happening and correlates analysis, and those data will emerge, I think, over the coming months and years. But I think some of the headline learnings are important to think about and have guided us in our thinking about our next sort of wave of investments and in terms of evolving our overall strategy. And the first learning, I think, is from the AMP trial, which, while it did demonstrate protection against sensitive viruses with high levels of neutralizing antibody titers, what it did teach us, on the other hand, is that the level of sustained serum neutralizing antibody titers that will be required for protection that was demonstrated in the AMP trial is actually significantly higher than what can reasonably expected to be achieved currently via the vaccine approaches that are in the pipeline. So that's a very, very important point that I'll build on in a moment. The 702 and 705 trials that were testing different approaches, of course, but with similar hypotheses, taught us that at least with these prime boost combinations, that vaccine elicited binding antibodies, so not neutralizing antibodies, but binding antibodies with effector functions, in combination with class 1 restricted memory CD8 T cell responses, that these responses are unfortunately inadequate to prevent heterosexual transmission, at least at a level of efficacy that would meet our minimum target product profile. And while there was some modest efficacy in the 705 trial, and that data continues to be analyzed and refined, we really didn't see any significant efficacy in the 702 trial. Next slide. So for us, this of course has generated a number of different and important questions to think about. But 3 key questions that we've really focused in on are first, given this challenge with the levels of neutralizing antibody titers that are going to be required for protection, can CD8 T cell responses potentially, in combination with neutralizing antibodies, reduce the levels of neutralizing antibodies that might be required for protection and get us over the hump in terms of what we might be able to generate with a vaccine? The second question is, can we develop simple translatable regimens leveraging the RNA platform or other nucleic acid platforms as referred to earlier, potentially alone or in combination with viral vectors or other platforms, to elicit sustained, and sustained is a key word there, durable and sustained levels of potent broadly neutralizing antibodies and durable CD8 T cell responses, including mucosal responses, so vaccines that are active at the site of infection. And the third question, which was also alluded to earlier, is can we really dramatically reduce the learning cycle time and think about ways to do faster, smaller, iterative human studies that are focused on answering specific questions, specific critical hypotheses, so that we can test and learn and iterate and test again and move forward quickly to refine our thinking and our learning over time. Next slide. So some of the ways that we're thinking about this now, and so in terms of the first question around can CD8 T cell responses reduce the level of neutralizing antibodies required for protection, we've been thinking a lot about some work that came out of a publication that Bali Palendran and his colleagues published from the non-human primate model that demonstrated that cellular immune responses could reduce the threshold of neutralizing antibodies required to confer protection. And so while we're thinking about this in a number of different ways, I wanted to highlight one recent investment that we made in some non-human primate studies with Scott Hansen, Louis Picker, Dennis Burton, and colleagues, to think about to test the hypothesis that a combination of cellular and humoral anti-SIV immune responses, each with partial efficacy, could synergize to yield higher efficacy. And so they're going to evaluate the rhesus CMV vector in an SIV form, which is known to elicit MHC-E restricted CD8 T cells, and combine that with either SIV trimer immunogens or SIV monoclonal antibodies to address this question. So this is in early stages, but we hope that at least in the non-human primate, this kind of experiment might help inform the answer to the question about CD8 T cell responses in combination with neutralizing antibodies. Next slide. In terms of the next question, which is, can we develop simple translatable regimens of immunogens? We are thinking about this as a potential to evaluate a portfolio of approaches and essentially combine immunogen design with various immunogen delivery platforms and, where needed, adjuvants for dose sparing or innate and adaptive programming, and then also potentially bring in some very novel technologies around sustained and pulsatile release. So in the immunogen design area, we and others are, of course, very invested in structure-guided B-cell immunogen design approaches. And I think Bart will elaborate on this more. So germline targeting approaches and other B-cell immunogen design approaches that are advancing quickly and very exciting and promising. But we're doing this in conjunction with leveraging George Shaw's SHIV non-human primate model to really try to understand how these immunogens potentially perform in a non-human primate model, and then also looking for opportunities to bring in novel CD8 T cell approaches. So in particular, Gaurav Gaya's work out of MGH, and approaches such as Betty Korber's with her mosaic T cell immunogen design, and delivering these, of course, via mRNA is a very promising opportunity. But not just RNA, but looking at, again, leveraging a long history of delivering CD8 T cell epitopes via viral vectors, and also bringing in other platforms such as nanoparticles. Adjuvants I already mentioned, which are really an issue more for the protein-based platforms, because the mRNAs are really self-adjuvanting with the lipid nanoparticle delivery. And then the sustained and pulsatile release technologies, which I don't have time to go into, are really about building on some data that suggests that if you give repeated and increasing doses of a priming immunogen, and then come in with a late boost, that you can really augment and broaden the BNAP response. But doing that in the clinic and obviously as a product could be extremely challenging. So are there novel technologies that might allow you to, that would release vaccine over time with only a single administration of the vaccine? So next slide. And then in terms of the last question, which is, can we reduce the learning cycle time and move faster and iterate faster? We, as many others, are trying to think about how we can move quickly into not only healthy volunteers, as we have historically, to characterize the immune responses from different vaccine approaches using laboratory assessment, but also thinking about opportunities to evaluate vaccines in people living with HIV in conjunction with acute treatment interruption, and evaluate in vivo functional activity and impact on virus load. And are there creative ways of working in both populations to iterate more quickly? Next slide. So I'm just going to touch briefly on the RNA vaccine platform, because I think Bart is going to go into this in greater detail, but I just wanted to make a few points, which is, I think sometimes when we hear about RNA in the press and people talk about it, there's sort of this sense that there's just mRNA is one thing, but mRNA is not one thing, and needs to really be considered both in terms of the RNA technology itself, the way it's delivered, the way it's formulated and delivered, and its manufacturing platforms. And there's many different opportunities and complexities in each of these areas that I think we're going to see exploding and evolving in the near and distant future. Next slide. So for example, in terms of the mRNA comes in different flavors, and the RNAs that we've all been hearing about in terms of COVID vaccines coming out of Moderna and BioNTech Pfizer are modified RNAs, but there are unmodified RNAs, such as the platform used by CureVac, and self-amplifying RNAs, for which there are a number of groups developing this approach, that all they're all different, and the immune responses that they generate, both in terms of levels of antibody responses and flavors of T cell responses and levels of T cell responses, are different. And so there's an opportunity to really look broadly and iterate and evaluate a number of different mRNA approaches as we go forward. And I think we're going to see, again, lots of opportunity to iterate and evaluate these in the future. Next slide. Just briefly, I think this is, again, just the beginning of a massive opportunity in the global health space, because RNA has actually been around for a little while. We were invested pretty significantly in mRNA prior to COVID, both with Moderna and BioNTech. But there are for HIV, TB, and there are other disease areas now that these companies are beginning to focus on, and the community is more broadly. And so there are massive opportunities to develop this platform for pandemic response and otherwise, and to improve the RNA platform, both in terms of dosing, manufacturing innovations, and in terms of the delivery systems, in particular the lipid nanoparticles that are used with these vaccines. And in fact, I just read this morning from Moderna that they have developed an inhalable lipid nanoparticle that could revolutionize the way they're delivering mRNAs for, directly into the lung for certain types of vaccines. So I think, again, we're just at the beginning of a revolution here in terms of some of the innovation. But there, of course, are challenges. So next slide. And I just want to, make this final point here, which is that in terms of I think there's a lot of emphasis being put on the benefits of RNA, which are significant in terms of speed. Our RNA can be manufactured quickly and really it can enable this rapid iterative translation of novel concepts. But RNA was not the only reason why COVID vaccine development went quickly. So traditional vaccine might take 10 years to develop. We saw COVID vaccines being developed in a year, but that was due not only to the ability to rapidly manufacture and test the RNA platform, but it was due to rapid mobilization of funding, rapid establishment of clinical trials, leveraging of existing clinical trials infrastructure, rapid enrollment of clinical trials, rapid regulatory approval processes, rapid mobilization of funding, et cetera. So all of those pieces came together to enable this incredible speed of development of COVID vaccines. And while we can leverage some of that for HIV vaccines, in particular the RNA platform and the speed with which it can be manufactured and evaluated, unfortunately, I think will be challenging to leverage all of the things that happened in COVID, given the sort of crisis nature of what happened there. So I think the next slide is my last slide. And I'll just end by saying that I think there's clearly a lot of opportunity in the HIV vaccine space, a number of challenges in the HIV vaccine space. And in particular, I think one of our challenges is that we are now in a period where there are, as everybody knows, a number of other HIV prevention interventions coming along in the pipeline, which of course is fantastic. Long-acting injectables, LA-CAB, lenacapavir, oral PrEP such as islatravir, but HIV vaccines will need to advance in this context and compete with this broader environment of other prevention interventions. So I think I will end there and hand it over to Ntando or Stacy. Thank you.
Great. Thanks so much, Nina. Just really such a beautiful sort of broad overview, and then some specific details on the mRNA platform. We do have questions coming in, but we are going to save those to after Bart's presentation. So I'm just going to hand it over to you. And just a quick introduction. I think you probably don't need much introduction to this audience, but next we have Dr. Bart Haynes, who's the director of the Human Vaccine Institute at Duke University, and he is going to talk to us in more detail about mRNA. So over to you, Bart.
Thanks so much, Stacy and Bill and Mitch. It's an honor to be here today. Just in the next few minutes, we'll focus on messenger RNA and lipid nanoparticles for induction of HIV protective antibodies. And just to start out with, what is the hypothesis of the Center for HIV Vaccine Development, which is an NIAID funded center that I lead? And our hypothesis is that induction of high titer and durable polyclonal serum, meaning many different kinds of antibodies, and mucosal broad neutralizing responses that will hit the conserved sites on the HIV envelope to multiple of these sites, called multiple HIV epitopes, will protect against HIV transmission. That's our hypothesis. What are our goals for the next 4 years? The goal is to achieve proof of concept that a broadly neutralizing antibody vaccine can be made. Now, Nina has very nicely given the history and up-to-date notion of where we are with regard to the non-neutralizing antibody trials. And it's the failure of the 705 trial and the 702 trial that has really spurred the interest and the realization that, as well as the AMP trial that Nina mentioned, that we're going to need a broadly neutralizing inducing vaccine. Our criteria, and we have a target product profile, and these are just two of the points of that TPP, is that we want to induce at least one type of broad neutralizing antibody to achieve greater than 50% serum breadth against difficult to neutralize, R5 tier 2 means difficult to neutralize transmitted founder viruses with antibodies that bind to a broad neutralizing epitope in monkeys or humans. And we want to induce durable serum and mucosal broad neutralizing levels for a minimum of a year in monkeys or humans. So what are the challenges? Why don't we have an HIV vaccine that induces broadly neutralizing antibodies? Well, complicated subject, but there are four main points. First is that the envelope, the target of the broad neutralizing antibodies, is covered in sugars. You frequently may hear it's a candy-coated protein. They're very heavily coated in sugars. And these sugars shield the glycoprotein from the immune system. There's immune tolerance, is that the body really doesn't want to make these kinds of antibodies readily. Otherwise, we would have been done many, many years ago. Rather, in contrast to the SARS-CoV-2 vaccines, where the body is quite happy to make very potent, broadly neutralizing or neutralizing antibodies that can become broad, the broadly neutralizing antibodies are disfavored by the immune system, and that the immune system thinks that the broad neutralizing antibodies should not be made. They're not harmful when they are made, but the immune system gets confused because of the nature of the broad neutralizing antibodies. Third is that the need for what we call improbable mutations. And this just means that when broad neutralizing antibodies develop, they have very high levels of mutations or changes in their amino acids, their structure. And many of these critical changes that turns antibodies into the broadly neutralizing antibodies very rarely occur. And so that's one of the reasons it takes years when broadly neutralizing antibodies do occur in individuals who are infected with HIV. Is because these are simply rare events. So part of the HIV vaccine effort is to turn rare events by vaccine design into common events. And finally, the easily made antibodies are not broadly neutralizing antibodies. They're the non-neutralizing antibodies, but when they are made, they tend to divert the immune system away from broad neutralizing antibodies. And so the non-neutralizing spots on the envelope can induce antibodies that outcompete the rare broad neutralizing antibodies. Now, this is just a figure of the envelope showing multiple sites that need to be targeted. It's just a visual representation. We need to have multiple antibodies against the same site, and we need to have antibodies against many sites in order to prevent escape. So, and all of these types of antibodies are not readily made. And so what are we doing to overcome this? This is a schema put together by Dennis Burton at Scripps and John Mascola at the VRC and myself. And basically it shows what Amina alluded to, is that what we're having to do to speed up the development of broad neutralizing antibodies and convert it to a common event is to design immunogens that stimulate what we call precursor B cells, or the germline B cells, the naive B cells that sitting around waiting for invaders to come into our body. And then instead of just leaving that alone, which for most vaccines, like the COVID vaccines, you just give one immunogen, one type of spike protein, and you get the kind of antibodies you want. We're having to design separate boosts to hit the next stage of the B cell after it starts off from a germline, and then a final boost perhaps that will take these antibodies and make them into what we call mature broad neutralizing antibodies that have great breadth, and also do it in a manner that they will be durable. And these are just the types of broad neutralizing antibodies that were on the previous slide, and I'll just mention a couple of these as we go forward. Now, AVAC wanted me to really talk about the relationship of the mRNA lipid nanoparticle technology to HIV vaccine development. There are two COVID-19 mRNA vaccines that have been approved by the FDA, as Nina said. They are very safe. There are no viral components other than the immunogen itself encoded as mRNA. There's no risk of genome integration. That means it can't get into our genetic material and mess things up. They're very highly immunogenic, means that they stimulate the immune system very well. That means low doses of mRNAs have been shown to induce protective immune responses to Zika, which I'll mention in a moment, to influenza, as well as to the SARS-CoV-2 viruses. And this work has actually been going on for many, many years. It didn't just happen in 2019. This is a big deal, ease of production. It's rapid and cost-effective large-scale manufacturing, which I'll mention in just a moment again. And so how does it work? These are the mRNAs, the little red squiggles, and they are encapsulated in lipid. The lipid can bind to the surface of cells and be internalized and get into the inside of the cells called the cytoplasm. Inside of the cytoplasm, the conditions change and these lipid nanoparticles break up, and the mRNA, again, the red squiggles, are released. There's the mRNA right there. And then in the cytoplasm, this mRNA undergoes what's called translation, and it encodes the sequences that give rise to the protein that is in the normal protein that's in all the currently approved vaccines is a protein for protecting against measles, mumps, rubella, and what have you. What mRNA does is that it's the gene that goes into your cells and tells your cells which vaccine to make or which protein to make. In the case of the COVID vaccines, it's the spike protein of SARS-CoV-2. And then that's what's recognized by the immune system. So what are these advantages? Well, a lot of this work was pioneered by Drew Weissman at Penn, and I'll show you just one couple of slides of data that these mRNAs can induce durable antibody responses. We are not sure why they're not durable enough for the COVID vaccines or high enough titers. They induce high levels of what's called T helper cells for antibody production, and that's one of the bits of magic. That's why they're so good in inducing neutralizing antibodies for COVID vaccines. They're practical for multiple component vaccines, and as I told you, we're going to have many components in the HIV vaccine to get a number of types of broad neutralizing antibodies to develop. They're much more cost effective than making proteins for each one of the components. And most importantly, it's the short production time compared to proteins and live and inactivated viruses. Therefore, for our job in developing an HIV vaccine, we can test many vaccine candidates much faster. But as Bill Snow said, the key issue is that mRNAs are not a panacea for the HIV vaccine effort, and is not going to rescue the 25 or 30-year effort that we've had simply by being a platform, because the key issue for HIV vaccine development remains in immunogen design, designing those immunogens that will move along these broad neutralizing antibody producing B cells to undergo the right maturation and make a rare event a common event. Another part of the problem is that the titers to prevent HIV infection are high. There's a study from the Vaccine Research Center where they did what's called a meta-analysis and looked at all the studies of antibody protection studies in monkeys. And they found out that CD4 binding site broad neutralizing antibodies, the titer for 95% protection was 1 in 529. For another region of broad neutralizing antibodies called V1, V2, the titer was virtually the same, 547. But for the membrane proximal region down at the base of the envelope, the titer is low, so that these antibodies in effect punch above their weight. And a lot of effort is being put into developing all three of these kinds of antibodies. Nina also mentioned the AMP trial, or antibody-mediated protection trial, that studied infusion of the CD4 binding site broad neutralizing antibodies, antibody VRCL1, 10 IV administrations over 72 months at 10 milligrams and 30 milligrams per kilogram. Overall, there was no protection, but for susceptible isolates, it was protective and gave us hope that if we could design the right immunogen to get the right kinds of antibodies, a broad neutralizing antibody vaccine could work. The serum titers needed to achieve 80% protection were, in this trial in humans, around 215. And remember, HIV is an integrating virus, and so we're going to need those titers to remain high in order to protect. In 2017, we brought Drew Weissman from Penn into our CHOP-D, and we did this study. We needed to know immediately, could the mRNA LNP technology that he and others were working on, could they work in monkeys? And all he had was the Zika mRNA. So we got permission to do that study in our CHAVI program at the time. And after one immunization, it protected. And then there's an award that was just given to this particular study. And this is true, giving the lecture in honor of the award at the award ceremony, and making the point that this was the first manuscript published that used the nucleoside modified mRNA lipid nanoparticle platform, particularly in monkeys, and showing protection. And this manuscript convinced BioNTech and others to take up this technology. It raised the question of how durable will mRNA-induced HIV antibodies be. We were able to keep these animals for a year. And so the green line here just shows that after one immunization, the responses were, the neutralizing antibodies were durable. We also immunized these animals with HIV, encoding, envelope encoding mRNA, and got neutralizing antibodies that were similarly durable over that a shorter period of time. But the point is that it may well be that the titers are not high enough to protect either in COVID or in HIV, and we'll have to do some more work to understand how to harness this aspect of the mRNA technology. So the issues for encoding a complex trimer immunogen with mRNA LNPs, broad neutralizing immunogens must be well folded to express these complex trimer immunogens. And we must define stabilization strategies of well-folded envelopes that result in the majority of mRNA encoded molecules being well-folded so they induce the right kinds of antibodies. And the short version is we've done this. Paper was just published a couple of weeks ago in Cell Reports, in which we looked at how do you stabilize trimers, the sequence of the mRNAs, such that when they get into the cell, they give well-folded trimers. And this is mRNA encoded well-folded trimer right there. Electron micrograph image. What about envelope scaffold nanoparticles? And here's mRNA encoded scaffold nanoparticles. And we have also shown the way to stabilize the mRNA induced proteins so that when they get inside the cell, they induce the full envelope as a cell surface molecule. So just two quick examples before we wrap up. The membrane external region where the neutralizing titers are a bit low. We have just finished a human trial funded by the Gates Foundation and the HVTN and the NIH on called HVTN-133, where we started off precursor lineages for a type of broad neutralizing antibody against the base of the GP41 of the envelope. And we could actually get a little bit ways down the lineage such that we actually had some neutralization against wild type virus. Could not get to the end, and so we're designing boost, building upon this trial in 133. We're designing primes and boosts that are in preclinical development as mRNAs, because we can do this much faster and iterate much quicker in order to get to the clinical trials quickly. The second example is what's called the CD4 mimic classes of CD4 binding site broad neutralizing antibodies. These two are called the VRCL1 class and the 8ANC 235 class. Forget about the jargon here. The two classes are very broad and potent neutralizing antibodies that bind at the same site and bind just like CD4 binds to the envelope. The VRCL1 class uses a rare light chain. That means it's one of the reasons it's a rare event. And we've all heard of the IAVI G001 trial. That induce precursors of the VRCL1 lineage. And I'll just show you one example of inducing the 235 class that uses many light chains, so it's less uncommon, more common. And the key is to design immunogens that can initiate and complete the development of these classes to their full potential. And this is for the CH235 lineage. All this is a cryo-EM structure of an antibody induced in a macaque with a germline targeting immunogen. And it just shows the antibodies binding to the trimer in dark blue and light blue, and the envelope is in the middle in light and dark structures. And what this basically shows is that it's binding just like the broad neutralizing antibody binds and just like CD4. So it proves that we've induced precursors in non-human primates. And this particle and envelope is now just about finished with GMP manufacture, and will go into the HV10 trials and we're excited about this trial working. The boost has already been produced GMP, and it's already worked in mice that are humanized. And so we'll have a prime boost to go into the HVTN very soon for this type of antibody. And so as far as mRNAs are concerned, in order to speed up getting to the end of the lineage, as with all of our lineages, we're converting over to mRNAs as full envelopes to be compared with proteins, and the boost are being developed in our CHAVI by a team of biophysicists and structural biologists with structure-based design. But what about the timelines? This is one of the biggest things that mRNAs can do. They can really help us move quickly. So the preclinical down selection is being cut down to about 10 weeks. GMP manufacturing for proteins is like 2 years. We're looking hopefully at about 5 months or less. We have a dedicated team of regulatory, process development, analytics, and manufacturing staff for each product that are being made at Duke. We have a GMP facility where we're doing this. We have 2 teams, 6 people on each team for each one of the CHAVI products, so that we can have overlapping product development, such that 2 to 3 messenger RNAs per year will be tested in the HVTN small experimental medicine clinical trials that Nina mentioned that the field is going to. Now, this is one of the slides I showed in my scientific advisory board, and these are each of the tasks that we're trying to accomplish in order that we believe we have to accomplish to have a functional vaccine. And we're trying to make all these stars green within the next 4 years. And this means that we can start off precursors, and we can get to the end of the lineage, which we haven't been able to do yet. We have, and durable responses are very, very important, and so on. So finally, this is just our manufacturing facility. We have 5 suites, so we believe that the utilization of these 5 suites for to make mRNAs is going to really speed the task considerably. Now, this is a question for the discussion, and I'll give my answer. And I think a critical question, given what we've learned with the COVID vaccines, is will people care about an HIV vaccine when we get to that point of deploying it? And are there countermeasures to this issue or not? Countermeasures being what Nina showed, is there other alternatives that the vaccine's going to have to compete with? I'm an optimist, and my answer to this question is yes. I think people will care very much about a vaccine if the preclinical and clinical data on efficacy are compelling and the safety data are convincing. And I think people will care about the vaccine if it's affordable, if it's available, if the immunization regimen is practical, and the vaccine's protective effect is durable for at least a year. But as Nina said, that's only a very, very minimum. We want it to be durable for longer. I acknowledge all of the terrific funders who have been steadfast in funding over the years. The folks at Duke, Harvard, and Penn who get involved in the mRNA work, clinical trials team, and I'll stop there.
But thank you very much, and I really love the answer to the last question. People will indeed care about the vaccine, and of course, affordability, availability, and of course, the rest that comes with it will really be important as part of that. And again, thanks to all our speakers. Who really have, I think from Nina, who's really given us a broad overview in terms of the bigger picture where HIV prevention comes into. And of course, we hear about the wonders and the science of mRNA, and really where it will take us and how it will take us into finding an HIV vaccine that is affordable and is available, and of course, that is protective. So this is the time where we really come to your questions. And of course, unfortunately, we really have a few minutes, but we are happy to go over time really to continue with this conversation. And I hope everyone is able to do that, because this is important. Otherwise, if you're not, as we have heard, the conversation continues next week and the week after, because we're really talking about a lot of P's, platforms and pipelines today, processes and prospects. And of course, you can add your own P's. You know, there's a lot of potential from what we've heard, possibilities are there, and of course, partnerships are very important. And of course, this is all about prevention. So I think we will take, please do like Jim has done in really raising the questions, and we'd probably like to give that an opportunity. I don't know if Bart and Nina, you've seen Jim's question. Well, the first one has been answered in terms of a high efficacy in terms of a vaccine getting approved. What does that look like? And as you know, Jim feels that Nina, you have responded in part, but please feel free to go ahead and really answer to that. But another question is, how will the field message an approval of HIV vaccine, say at 50 or 70% efficacy in light of COVID vaccines that are 90 and above? You know, have we trained the world to expect efficacy that is higher than 90, and especially through this process? And will communities trust something that is significantly lower in efficacy in comparison to COVID vaccines? I don't know any of you who's really happy to take on to that. Otherwise, we encourage everybody can use the chat box and use the question and answer so that we can really take on a few questions with the time that we have left.
Well, I think one of the keys, Ntando, is that, and Jim, that HIV is an integrating virus. And that means that we don't have a chance once the virus is integrated, which takes 3 to 4 days after initial contact with the virus, to really protect. And once integration occurs, then it becomes invisible to the immune system. And that's quite different from the COVID vaccines. I think the 50 to 70% efficacy comes from the modelers who say that even with that kind of vaccine, over time, it will abrogate the transmission rate in the pandemic and have a significant effect, although not completely. But I think it really is a very important question that the expectation set for the general public by the COVID vaccines is incredibly high. And we already know the bar is incredibly high for an HIV vaccine. We can't fail to prevent transmission, whereas the COVID vaccines, their mission and all other vaccines really is to prevent serious disease and death. And we have to prevent transmission with an HIV vaccine in order to do that.
I think I'll just add that, I mean, it's a really great question, Jim. So thank you for asking it. And it's really about what the acceptability is going to be at the end of the day. And that, of course, will vary based on whether we're talking about, what region of the world we're talking about in part. And the bar has been set high with COVID vaccines. But I think the bar is also being set high with other prevention interventions, as has also been mentioned in the chat. And so I think that, in the near term, our goal needs to be to get to some reasonable level of efficacy with an HIV vaccine that we can build off of and introduce vaccines as an option in a broader toolbox of other options, so that people have choice. But I think and ultimately, to get to a vaccine that will be attractive to people, we do have to keep in mind that it's going to have to be highly efficacious and durable and simple. I don't think we can emphasize that enough. And unfortunately, we're pretty far away from concepts that will meet that bar, but we're still learning. And we have to take baby steps before we get there. But we will ultimately need to come up with a vaccine that can compete with other interventions that are increasingly appealing to people. So yeah. I'm also an optimist, but I'm also a realist. You have to be an optimist to work in HIV vaccines.
We have to come with a vaccine that has to be better than what we have.
Yeah.
So, I mean, we and people are really excited about the P's that need to be considered in the process. Angela is talking about price as one of the P's. Khadija is talking about people, which I think is one important aspect that we probably, in the excitement of science, you have to remember all the time. And Stacey, we are right at the top of the hour. What do you think? Do we have more to continue?
Well, maybe just very quickly, I think just to kind of bring it back to this specific call and the topic of platforms, and I think this, I think Nina and Bart, you both spoke so well to this issue around mRNA that I think is on all of our minds, that it's really sort of come into the limelight with COVID, and now is being seen a little bit as kind of the sort of the answer or the silver bullet for HIV vaccines. And I guess I wanted to sort of dig into that just a little bit specifically. Again, you both sort of spoke to this, but just wondering, I mean, can we kind of say that there is a kind of a consensus in the field that when we think about this issue that we're looking at right now with HIV vaccine research specifically, and this need to kind of iterate much more quickly than we have before, is mRNA kind of the best platform? Do we have sort of confidence that mRNA is the best platform for that? Can we kind of say that there's consensus around that? And if we can, is there sort of is there potentially a risk of looking so specifically at mRNA?
So I guess I can take a first crack at that. I think there is a fair amount of consensus about the utility of mRNA as a tool for all the reasons that we've talked about in terms of the pace at which it can be manufactured and the ability for mRNA to help us iterate faster and learn faster and shorten the cycle time. But I think we've distinguished between mRNA as a platform and the immunogens that are expressed via mRNA, and think of really, mRNA is really the software, right? And it's the sequences, it's the immunogen design ultimately that will determine whether or not we have really good vaccines via mRNA. And so I don't think that we're putting all of our eggs in one basket by necessarily using mRNA as a key platform, because we're going to be needing to evaluate a variety of different approaches as it relates to immunogen design. And I also think that, as I said in my talk, we're going to need to think about combining RNA as a platform with other platforms and not singularly focusing on RNA. Because it may be that we need some combinations to get to optimal responses. We don't know yet the extent to which RNA will be optimal for CD8 T cell responses. I think there's some hint that it can do pretty well. But we also don't know whether or not RNA will get us there in terms of durability of responses. So I think it's a fair question, but from my perspective, it's really more a tool to help us iterate and learn faster in the near term.
Yeah,
do you agree with that?
I totally agree with everything Nina said. The only thing I would just add to is that, and deeply embedded in your question, Stacy, is okay, so it's going to clearly be the platform for us to iterate. But are we going to lose anything in iterating with mRNA that if we'd done it with protein or some other platform? And I think there are data now. We've done comparison studies, and the HVTN has several trials planned to compare head-to-head mRNA and protein, so we make sure we don't make that mistake. But the data that are published so far suggest that, or have been presented as well, suggest or show that mRNA is equal to protein. That is, we're not losing anything in immunogenicity-wise. And then as an immunogen to get around some of these roadblocks that I talked about, mRNA may actually be a little bit better. It's the durability that we're stuck on right now.
Yeah, and so I can just add to that. So there isn't, an ongoing trial, the G002 trial that's currently with the Moderna platform and the IAVI NAC EOD vaccine that's doing exactly what Bart just indicated, which is comparing RNA to protein. So I think that we will get answers to this question very soon, but we have certainly not given up on using other platforms, and certainly expect to continue to look at nanoparticles, viral vectors, and others as well. So
great, thanks for that. And again, I think really nicely pointed to in both of your presentations, but super helpful clarity, and I think good things for us to be watching in the coming in the coming years. I know we're over the hour, and there were other questions that we wanted to ask, but I think that what we'll do is I think use this as a platform for the series moving forward. Again, we're really hoping, especially in the sort of the third webinar, looking at kind of prospects, we'll bring back in some of these themes. And just again, I mean, from myself and Ntando, did you have any final thoughts?
I know, perfectly
Okay,
good.
great,
I think
great,
we're
great, great.
looking forward to the next one, which is processes. And I think it's talking more, you know, as a lead, basically adding to the conversations. And there's always a lot more to talk about, but I think it was important to really highlight these, especially on this very special day where we remain hopeful about the possibility of an HIV vaccine.
Great. Thanks so much. Bill, any final words from you, our chair for the series?
Nope, I just said goodbye in the chat.
Okay.
Have a wonderful day, everyone. Bye-bye.
Okay.
Thanks so much.
Thanks so much.
Thanks,
Bye all.
everybody. Bye-bye.